Thank you very much for the invitation. It's been a pleasure to be here. Now I'll set my watch to 42 minutes so I don't get yelled at. And then it will make a lot of noise at the end. I apologize for it in advance. You guys are very tired. You've been here for a long time and you're like, I got what I wanted. I want this to be done. But there is more. So sorry. And this one is not going to be particularly fun or easy to understand, because what I decided to do, I will talk about something that we haven't even published yet. But this is science as it goes, and I thought this would be a good opportunity to get opinions from people who study population genetics, because I have no idea about population genetics. I'm coming from a very different route. I did, when I was young, I did cryptography, operating systems, and I'm coming from the hardcore, dark depths of computer science and the things that we don't want to remember anymore. And then I moved on to machine learning, signal processing, and I did nanopore stuff for a little bit. And then I jumped into microbial ecology. And then as the time went by, I started realizing that there are some very interesting questions in microbial ecology that we cannot really study through marker genes, marker gene amplicons, which what I was doing at the time. And so I started putting more and more attention to metagenomics. And I realized we were missing a lot of things in our toolkit to study environmental omics data that can get extremely complex, as you all know. So we started implementing this platform. So first I'll tell you a little bit about the platform before I jump into the scientific question we're interested in and what we have been developing from scratch to get closer to some meaningful understandings of it. So the platform we have been working on is called Ambio. It's new, so you will not hear about this very often, but it has about 40,000 lines of code, and it's distilled from 200,000 lines of code. So we, instead of writing ad hoc scripts, we created a framework, a very solid framework that supports from studying comparative genomics to metagenomics or metatranscriptomics. So it's a unified core, and we're adding tools around it now. It has about 100 programs. It's like a Lego. You just build it, and it's painful. But it's supported by online tutorials. We write a lot, more tutorials than papers, to be honest. When we are out of this field, we will be like, yeah, well, we remember with our tutorials rather than our papers. But we do that so you don't need us, not you or microbiologists in general. How many of you are microbiologists? How many of you identify yourselves as microbiologists? Tito. It's it. Okay. Lovely. So I'm talking to you today. Okay. And the rest just try to follow. So we believe that computer scientists should not be the leaders of this field. There's a lot of data, yes, but the inspiration should come from, in scientific questions, should come from microbiology or biologists, life scientists in general, I think. So we are trying to make sure that they don't come to us. They do their own stuff with these tutorials. That's why we put so much effort. And also, Ambio everyone has a platform, but how many of the platforms is a cross-stitch art, done by someone who has nothing to do with the platform? So people are very supportive of our stuff, and they do whatever they can to help us feel good about ourselves. And Ambio is, in a way, a radical departure from the current software development practices, and it drives you crazy, both with joy and frustration. So it is not easy to learn. There's a steep learning curve. And it's not really, even for me, it's not easy for me to wrap my mind around the code base. How do you make sense of 40,000 lines of code, made up mini modules and hundreds of functions and classes? So I did this. Now Ambio can introspect itself automatically and create this network to show how concepts are linked to little programs, linked to databases, etc. So you can look how a bunch of context data put together as a context database, and then how other programs could operate on it, and how it's linked to the concept of profile databases in Ambio, etc. So it's not really easy to learn. I know I'm not really doing a very good job selling it to you, Tito, but it really makes you able to study things very in depth, because it's like a bunch of Lego you just piece together to do things that we couldn't envision. And also, it does all these nice visualizations. Everything in Ambio turns into an interactive visualization. All these figures are screenshots, but you zoom in, zoom out to these, you right-click, you click on things, you inspect at the gene level coverage, you look at codon level things, you can overlay environmental variants on tertiary structures of predicted structures of your genes, etc. So there's so much you can do, but trade-offs. I mean, nice visualization, very steep learning curve, so you make your mind. But when people made their minds, we try to help them by giving workshops. These are some workshops we have been giving around. This is from France, cheese and wine. And so we try to help people. I give these very intense 2 days workshops, like 10 to 12 hours both days. And people either like me at the end of it a lot or they hate me. You know, everything comes together, the accent and style, and they're like, okay, I develop allergies now. But okay. So the reason we're been developing Amio is not because we're interested in tool development. I, in fact, hate tool development. I want to be done with it. But, you know, you come up with questions that require you to do something about it. And we don't always have the right tools to study these complex questions. And then, unfortunately, tools have become ways to define how we are even asking our questions, if you think about it. A tool emerges, it's very useful, it's great, and everyone starts to design their experiments knowing that the data I will generate from my experiment will be useful with this tool. And then there is this stupid feedback loop starts to emerge where we start designing our scientific inquiries based on what's available. And so that's why Amio was an attempt to break that to a certain degree, because it gives you nothing but pieces of a Lego. So if you know your stuff well, Amio will be amazing in your hands. If you don't know, it will give you nothing except silly visualizations. So what do we do with Amio in our lab? We have multiple different trajectories. I will summarize just 3 of them, and then I'll talk about one of them because it's most relevant to you, I think. We're very interested in understanding microbial diversity and ecosystem functioning. So we do metagenomics. We recover population genomes from metagenomes, and we are able to curate them very extensively. And then we, through genome-resolved metagenomics, we talk about how environments function. Recently, for instance, we recovered about 1,000 genomes from the surface ocean, among which we found nitrogen-fixing populations. I know you don't care, but very important process in the environment. It's like primary productivity of oceans. It's kind of a bottleneck. Hello, people. You want to come in? No? You don't? There are some people there you don't see. That's okay. So, so we found organisms that were capable of nitrogen fixation occurring orders of magnitude more abundant than the last 30 years of estimates. So when you look at these at this level, you recover fundamental insights into how the Earth works, how microbes make the ecosystem tick. So we're interested in that. We're very interested in determinants of microbial fitness. How can we identify genes and genomes that are likely to contribute to the fitness of microbes in the environment? Can we really tease them apart, etc.? For this, we're linking now pangenomes to metagenomes. So we're taking a bunch of genomes, put them in a phylogenetic context, understand how gene classes look like with respect to each other, and then we dip it into metagenomes, come back and say these genes are linked to the environment. I'll show a very quick example, I think, of a metapangenome. And then we're interested in microbial evolution and population genetics, but through single codon and single amino acid variants. And that's the concept I want to talk to you about today, because we're just in the process of developing this, and we don't know what we're doing, basically. Probably you guys already know much more than we do about some of the things we're doing. Okay, so if I focus on this for a while, I need to thank to this excellent project, Tara Oceans. You don't know, but these people went around the world, collected all these samples from the ocean, surface ocean, and different chlorophyll A max, and different depth profiles of the ocean. By the way, no one talked to you about oceans, right? I'm the only one. Good. Microbes occur in oceans too. I mean, we're all driven by medical questions because that's where money is, but I think ocean ecosystems are excellent places to study evolution. So that's why I'm going to be an ocean guy today, although I know nothing about oceans either. So Tara Oceans project took a yacht and a couple of dozens of scientists, and then they traveled around the world collecting samples, filtering water, and creating biomass from different size fractionations. So we have bacteria from surface ocean and diatoms and things like that for different depth profiles as well. And Genoscope, an institution in France, sequenced the crap out of this data. So we have now this very high quality data sets coming from different parts of the ocean. So we can look into microbial makeup of these places. And there are many institutions were involved with this project. And they did, I think, this is one of the most significant resources, metagenomic resources in the world today to study oceans. And I'm very thankful to these people for being able to come together and do this. So the project I'm going to be talking about now is intellectually and technically led by a couple of people from my group. I wanted to thank them in advance. So if I don't have time, if my alarm goes off, I don't miss to mention their names. So, okay, what's the question? The question.
Oh.
Yes.
What's your question or our question?
What's your question? Tell me.
Okay, my question. So first, my question was whether you are interested in ocean, and you said yes. But this boat is the boat of Venter.
No. Venter is he's not doing boats anymore. He's doing faces now. So.
Oh, really? Okay.
Yeah, he's Venter. No, this is another boat, but this is new. This happened just recently, and it's more of a public effort. So European institutions chime in, and they created through very tough processes. They gathered enough funding. Now they're continuing. So this is more of a public effort. Any other questions? No. Okay. So the question I'm interested in: how did the genomic heterogeneity emerges within a single population? Right. That's the main question, and more importantly, maybe how does it drive the partitioning of ecological niches? So, just to make an analogy, I want to give you this. Just imagine the ocean, this huge thing that goes around due to oceanic current movements, like your populations going around on Earth. And if I'm standing in front of Woodsall on this rock looking at the Atlantic Ocean, and I'm holding this one microbial cell and just drop it in this otherwise sterile ocean, okay? And this one cell goes in there, and then in about 500 years, this body of water comes back in front of me again. That's how this works. Oceanographers have figured out how these eddies and currents are working around the ocean. And of course, when this guy comes back, it's not alone anymore. There are many cells that ancestrally linked to this one cell. And then, of course, these are not identical to each other at the genomic level. So the question is, because polymerase makes errors, and then due to multiple different evolutionary processes, some of the variants gets fixed in the population, some just gets lost, some of them positively selected, some of them are there just because they are doing their genetic hitchhiking. But at the end of the day, we have all these cells coming from the same population with slight variance, differences. So would you expect, due to only given the oceanic current movement speeds, would you expect ecological patterns to emerge if there is no selection, even? Let's just think about this as a thought experiment. There is this, in fact, this is what people think about a lot in our field, because we see, when we look at the oceans, we see different microbial populations in different parts of the ocean. And how did this happen? In a constantly moving body of water, how do you expect such ecological niches to emerge? One paper suggests that biogeographic patterns in ocean microbes emerge in a neutral agent-based model. So this is an amazing paper by Hellweger et al. And we find it very insulting. That's pretty much what everything you'll see is discussing findings in this paper to a certain degree, to contrast with that. They suggest you can take a couple of genomes, implement a neutral agent-based model, so there is no selection, and you can just follow oceanic current movements and let these guys go around the ocean. And when you run a couple of million simulations, generations simulate there with a given error rate of the polymerase, you would start seeing patches of the ocean looking more like each other. I guess this is no surprise to anyone. It surprises you. No? Yes? Do you want me to try again?
I think that in the ocean. Go
on. Okay.
I think.
All right. Fine. So this is a simulation, and it's a lovely one, very smart one. But it is truly a challenge to study the influence of evolutionary processes in practice. Because so our advantage is this: how many population genetic data do you have from humans? Like, what's the largest data set we have? Can someone tell me? What's the largest data set we have right now from humans to study population genetics? Million people? Okay, let's say 7 billion people. Let's say we had data from every single individual on Earth. Well, 7 billion cells is about like a glass of ocean water. So we have that many bacterial cells there. And how long humans live? They live a lot, much longer than bacterial cells. Reproduction, the speed of the turnover rate of the population is like a day in the ocean. So phages destroy these populations and then they come back. And just imagine the rate of reproduction, the amount of genetic information that is just going crazy. And there are many glasses of water in oceans, and many of them are surface ocean too. So we're looking at this crazy amount of evolutionary interplay that is taking place. So, but of course, the main question is, how does the genomic heterogeneity emerge at the first place? That's the beginning of speciation, right? How do these things work? But we need to understand, we need to be able to tease apart these questions given the three tremendous needs met. The first one, you need a population that's everywhere, right? So we can start looking at geographical partitioning of oceans in the context of the single population. Like you have humans, great. They are everywhere on Earth, so now we can look at how Earth is being partitioned by humans, and you can predict how they move around, etc. So in the microbial world, we need to find an example to that. Two, we need data. We need crap loads of data and very deeply sequenced metagenomes, because nothing is very abundant in the environment when you look at microbes. And I'll show you one example. We have one. And three, you need a proper way of analyzing this complex data that could tell you about non-synonymous variants very accurately. And we don't have that. And I'll show you why. This surprised a lot of people, but you cannot read from metagenomes and talk about synonymous versus non-synonymous variants, period. And I'll convince you to that in a second, I hope. So we turned our attention to SAR11. SAR11 is a microbial clade that is composed of many genomes, thanks to some people who have been isolating them, including Steve Giovanni and Mike Rappe, from surface ocean. And these guys are extremely abundant. They make up to 30% of the surface ocean samples. Just imagine, 30% of that billions of cells. It's just tremendous amount of population sizes. And there are cultivated, isolated members of these SAR11s. And what I'm showing you here is an amino figure that shows the pangenome of all isolates that was available to us at the time, which is like just recent. So every concentric circle is a genome, and every spork, or whatever we call them, tip of that dendrogram, represents a gene cluster. And the presence of a mark here indicates that particular gene cluster, this genome, contributes to that gene cluster, at least one gene. I hope this makes sense so far. Does it make sense? Who is completely lost? Okay, so what we do basically is this. We have all these genomes independently. We identify open reading frames in each one of them. And then we do blast all. We blast all open reading frames to all open reading frames at the amino acid sequence level. From this blast comes associations between this amino acid from this genome and this, this gene from this genome and this one is this identical. And then this. There are hundreds of millions of lines of entries like this. And then we turn this problem into a network representation, and we create a network. How these genes are linked to each other. Imagine if there's a gene in every genome that's almost identical, they create this cloud of genes that are all linked to each other because they are identical, right? Because they hit 100% blast. So this gene is linked to this gene with this genome, and this gene is linked to another gene from another genome, etc.
The network is over genomes or
genes? Genes. At that point, you don't know genomes. Exactly. Just looking at genes. Exactly. And then you ask the MCL algorithm to find you meaningful groups in this network. Like, can we find clusters? I'm not going to go into details, but MCL is a very good algorithm to do that kind of stuff. If you're familiar, if you're not familiar, take my word for it for now. Then you have your gene clusters. So you find those things, and then it becomes a thing over there. And then you ask the question, okay, I have this gene cluster. How many genes do I have in it? For instance, for some, you have only one. And then it turns out to be like this guy here, let's say. Because it's coming from this genome, but none of the other genomes had anything to contribute to the gene cluster, because in the sequence space, it's so far from everything else. And then you take another gene cluster and say, okay, how many genes do you have? It says 21. And then it's somewhere here. So every genome contributed one or more. to that. So this representation tells us which genes are core, which genes are extremely similar across these genomes, which genes are singletons, which genes are more relevant to maybe ecosystem functioning, accessory.
So how many nodes are in this graph?
How many nodes?
Yeah. So
it changes. As many of these forks here, of course. Well, final clusters, yes.
And all the genes have to be like represented in some kind of known database? Or
No. This is completely independent of databases.
just blast of every two
Exactly.
genes. So
Yeah.
sequencing,
Yes.
compare the sequencing.
Yes. We can talk much more about this if you want later, but the only point here I want to make is that I'm going to focus on that one red guy, and I want you to see that it's not extremely different than everything else. When you look at in a phylogenetic context, a pangenomic context, it doesn't stick out. And those organization of those genomes are extremely similar to what the literature tells us about their phylogeny. Independent metrics also confirm that. Those subclades are coming from literature. So fine, so far so good. So what we do in the lab, we take this pangenome, we literally dip it into the ocean and bring it back, trying to understand how much reads do they recruit from various environments. Okay, that's a way to link this dry, boring information to what's going on in the environment. So for that, we use ocean metagenomes. They are coming from all these sampling stations. So and this, I like this because this is also is known as, this is how you do it, US. Europe tells us. Because in the US, we're very good at generating data sets of total irrelevance in the field of microbiome. Don't tell those big names you're working with. I really like them, and I hope they write me a good letter. But basically, what we can do using this tremendous amount of data that they generated, we can take this pangenome and put it in the context of those metagenomes, and then we get this over there. Okay. So that is summarizing 36 billion sequencing reads, that little heat map. And what it shows us is where do we find these genomes recruiting reads from metagenomes. So I know this is a very kind of non-intuitive concept because you never do this pretty much. But I will try to give you an understanding of it very soon. But maybe I should start with this one. Those little squares, so every line there is pretty much coming from one of these things, right? For instance, it's this one, because metagenomes organize by latitude. We try to organize them by latitude. And for instance, that sample corresponds to that particular line, that row. And let's say this sample is that one and that sample is that one. I hope that it makes some sense. And you can see one clade over there is completely bipolar because those are the ones that like colder waters, okay? And then there is this temperate zone here. And this part here in this display is like all coming from things that are not so bipolar. There are more temperate oceans. So what sticks out from this figure immediately is that there's this one dude that takes up the entire goddamn ocean because it's recruiting tremendous amount of more reads compared to everything else, despite the fact that there are no major differences in the pangenomic context. Like, why this guy is so abundant is a question that I will not answer because no one knows. But now people are interested in this because in other labs also people have been realizing that this particular isolate that's coming from Hawaii, and I asked Mike Rappi to take me to the place he cultivated it, and I even took a video of him explaining how he cultivated this particular one. This particular isolate gives us access to the most abundant microbial population on Earth, given all the metagenomic data on all isolated genomes available to us today. So it's kind of crazy, I think. But we don't care. We care about the fact that this now is the one of those things that we were looking for, something that's everywhere, to study the question, what drives its evolution, or how does the genomic heterogeneity emerges make sense to us. Because as you can see, it's in all temperate ocean samples. So I will make this statement, and maybe we can discuss a little bit what it means, but maybe that will be enough to you. The isolate, HIMB83, is just a member of a large SAR11 consortium of cells in the environment. It gives us access to it. And it's just a member of it. And it's very critical. When you take a human member of all human populations, you don't expect this human to have a third arm in its back that is not really representative of what's going on in the environment. But with microbes, it happens quite often. We find like third arms and second heads and or no head whatsoever. And then you get a member, you manage to cultivate the third arm guy because your alien ship was, required that arm to be there to be captured from Earth. So you get a sample from Earth, but it's how representative it is of everyone on Earth. How could you understand that? You're an alien, you want to understand humans. You could do this. You could go back to Earth. You can't capture everyone, you know that, but you can chop everyone up. You could chop them up, sorry, into pieces. So now arms and legs and heads and everything is just floating around. And you could take every little piece. How many times do I observe this arm at this place in this human that I managed to capture? And then you would realize that the third arm is very rare. It never occurs in the environment. So you would say, okay, you know what? Third arms coming out of the back of people is not really a very good predictor of humanness. Like, it's not really a core feature of humans. So that's pretty much what we need to do with microbes, because it happens so often. They don't care. They don't care about how they look. They don't care about their shape. So which genes are there in HIMB83 that we can rely on to study its evolution? So that's the question. And yesterday night I realized that I'm not going to be able to communicate this very rapidly to you, because most microbiologists do not need to understand this part, but Tito knows, for instance, but you all don't know. So I decided to put together a very crappy representation of what I'm talking about using my iPad. So welcome to my attempt to describe this concept, finding core genes for you by linking things to environmental genomes. So this is your human, and this is an arm, this is a second arm, this is a third arm, etc. in the context of HIMB83 genome. What we do, we go to the metagenome 1 and recruit all the reads we can that are similar enough to this genomic context to map. Like a random piece of, limb you found on Earth, it looks like a human limb, so it kind of aligns here. But a deer leg is not mapping here. So because the ocean is filled with, you chopped up everything. You're terrible. So you have everything, you want to find human-related things. And this creates a lot of complications that people don't appreciate in our field today. But let me finish with this. So you look at the coverage of this genome in this metagenome. Let's say this metagenome is Earth, and then there's another solar system that we don't know about, but as an alien you know there are human-like things there as well, so you have other metagenomes as well, okay? So in this particular place, everything seems to be working well, every genome is covered, but you go to other metagenomes, other planets, and then you find that, well, there's no read recruitment there, and then you're like, okay, you know what, I'm not going to trust these things because they don't seem to be covered throughout the entire collection of metagenomes I have that represents the niche boundaries for this population to exist. So those genes are not the core genes. Does it make sense? Okay. So for instance, if you were to focus on one of those genes that you determined to be core, I even put the effort to put those blue things. I didn't have to.
Every such distribution is from the points you showed on the globe?
Exactly, yeah. So I'm showing 3 examples, but basically that's what it is exactly. So if we focus on that one gene, and look more carefully, you would see nice unified coverage of things. And now we're talking about, we're getting closer and closer to population genetics level insight. So when we have all these short trees mapping to the genomic context, as you know, we can ask the question, how many of them have differences from the reference at certain nucleotide positions? So basically, that's exactly what we do, okay, for the core genes. Now, first I will show you the core genes of HIMB83. So when I do exactly what I said, this is what I see. There are some genes consistently occurring across metagenomes, some genes that are completely lacking here of them because they are third arms or second heads, etc. And there are some genes that show geographical distribution patterns that we don't want. Those are accessory genes likely, okay? So from here, we can write a silly algorithm to find out things that are systematically occurring in all metagenomes to find those core genes that I tried to allude to, okay? When we have them, they do not contribute to this to distinguish different environmental sites from each other. If you think about their presence and absence, if you had a matrix and if you were to try to cluster all metagenomes, in an ideal world you would get a flat line because everything is everywhere and everything's, so all your matrix is 1, 1, 1, 1, 1, right, for each gene metagenome pair. Okay, so we're going to make sense of these thingies here, but how? For that, we're using single amino acid variants. So that's the kind of novel part that we're doing. And single amino acid variants are like single nucleotide variants, but slightly different because they focus on explaining the frequencies of codons and amino acids in the environment. and I'll show you why it's important. And single nucleotide variants are like the correct way of saying single nucleotide polymorphisms, SNPs, because in the context of microbiology, we can't use SNPs to talk about what people talk about elsewhere in science. It doesn't apply. It's just a poor nomenclature. But I don't make a very big deal out of it. And single amino acid variants are better than single nucleotide variants if you are interested in understanding non-synonymous variants in the environment. Because you cannot recruit trees from metagenomes and confidently talk about synonymous versus non-synonymous variants in the context of microbial life. You can do it in human stuff perhaps, but it's not going to work in microbial life. And let me show you why. Let's say we have this gene, and there's a codon, and there is a codon coding for serine. Serine can be encoded for one of those other codons, you know that, because you have the table. And then you go to your environment, you recruit your metagenomic reads, and they align underneath this codon like this, like those reads here. And where's my, okay, yeah. So what you do, you look at this position, and then you realize that not everything is identical. So you have A's and T's. You ask the question, are there TG, because A is synonymous already. You say it's synonymous, right? You tell me whenever you don't agree with me right now, because this is kind of important. And then you see there are some T's in the environment. Fine. Do I have any TG? Because if I have TG in the set of codons, then I would be like, okay, this is really it could be non-synonymous, right? It could be TC or other things, but you don't have anything that starts with TG. So you say this is 50% likely, 50% non-synonymous. You don't know, but you conclude that it's very likely that it's the case. You go to the next position. You have C's and G's. Do you have GT in the list of codons? Yes, GT here, these G's. So perhaps this is synonymous. You don't know, you're not sure, but this is very likely synonymous. And then you go to the next one, it's all T's. You're like, fine, I don't care about this one. So you conclude from this that there are likely some non-synonymous variants, but mostly it's synonymous, all the variation you observe in the environment by looking at that. What we do, we remove all the reads that are partially mapping to a given codon from the environment and get rid of those. And we use only physically linked nucleotide positions to start looking at frequencies. And then often we learn that this is, for instance, encoding for threonine, and this is encoding for cysteine, and this is in fact, all the variants are non-synonymous in the environment, 100%. And this is the only way to make this prediction confidently and 100% accurately. So another thing that our tools enforce us how we think. We take those pileups because that's what the tool does. We get pileups, it's very easy. So we keep using samples to get pileups, but it doesn't work. So we need to change this. It's very computationally expensive to find short reads from metagenomes that are mapping to the same codon as a whole. It takes us, instead of 3 hours, 3 days. But then we have absolute data that we can trust because these nucleotide positions are physically linked. Okay, so I want to show you one more thing because I'm going to quickly wrap up our results. But what happens when we do this for our genes? And this, for instance, if I were to take this gene and look at single amino acid variants, I learn about how those single amino acid variant frequencies change across environments. That's one insight. But there's one more thing I can do with that, and I think that's extremely critical, and I'll talk about it just a little bit more. But you can take those single amino acid variants and you can put it in the context of tertiary structures. So in maybe this gene, this particular tertiary structure, it looks like this when you overlay single amino acid variants on it in metagenome X, and it may look like this in metagenome Y. And this is not random, this is real data. And you can see how the solvent inaccessible part of the stem barrel protein is so fixed. And solvent accessible part of it is, in fact, very enriched with variants. So what we do basically is to use these variant profiles and amino acid identities to create a huge matrix of infinite amount of data and try to resolve it. Ask the question, how do metagenomes correspond to each other given these variants in the context of these genes? So forget about the structure now, because we're not using structure for anything at this point, but we will in the future. But basically, you get this kind of table for your genome across metagenomes, okay? And then it's like, okay, we have these metagenomes, we have these, for every codon position for every gene in every metagenome. We get these corresponding gene code. This is the codon order. The reference say it's an ASPAR gene, and the departure from reference is this much. Here's the coverage of this with short reads, and this is the actual counts data. Look how clean this is. It's asparagine or glutamic acid, not anything in between, except some small amount of noise. And then you know how likely to observe this kind of stuff happening by chance, and what are the competing amino acids, what's the consensus, what's the departure from consensus, what's the weighted estimates of Blossom scores, and then you can look at Kull-Black-Leiber divergence, raw or normalized, or entropy across samples. So you get tremendous amount of data, and it's pretty intimidating. And if you just think about this, how would you resolve this? I mean, you have, like in this case, for instance, we have one genome, 1 billion reads recruited by that, and it's about this many single amino acid variants. You could have codon position, metagenome, and a feature vector, right? What's going to be in your cell? Just let's imagine for a second. I have my metagenomes, I have my codon positions of core genes, and I have something in those cells, right, in my matrix. Those cells are 21-dimensional vectors, correct? Each of which correspond to an amino acid. And I have frequencies in that. So now it's already a little bit more dimensions than we wish to have for our machine learning algorithms, but then it doesn't end there. The question gets even more complicated because some of those amino acids are functionally homologous. So you may have something but purifying selection. It's just, it doesn't mean that there is positive selection going on there. So some of those, you can compare each one of them. You may have Blossom scores for those. And then it doesn't even end there. Some of those may be solvent accessible. So you can functionally non-homologous amino acid variants that are not necessarily indicative of positive selection or anything. But you have others that are inaccessible by solvents. So when you find glutamic acid going tryptophan there, it means something very, very significant. So this becomes a very, very complex data very rapidly. And we have a lot of it. And we have no idea what to do with it. And how do you link all this information to structure? We have it. We have structures. We have the structural makeup of most genes we have. Not most, some genes we have. And then we have all the space. We know how they move around. So now can we say how to tease apart? Can we get closer to teasing apart positive selection from neutral processes? So what we did, we used deep learning, auto-clustering output layer implemented by our collaborators. They are actual computer scientists, not like me, a fake one. So they have this very nice algorithm. In fact, this is a graph-based algorithm. This is neural networks, basically. But then, of course, we don't have labels. So they implemented another thing. So we could learn latent variables and come up with appropriate labels for an unbiased estimate of distance between our metagenomes given this data. And I'm not going to go into details of these. I couldn't even if I wanted to. But there are these papers out there, and we did our best to explain them in our study as well. Basically, what the patterns I'm about to show you are robust to pretty much everything. We can use stupid Euclidean distance and get the same things in the context of this population. So what we did then, we took all that data, and then we asked the question, how do metagenomes look like? How do they correspond to each other given all these things? And this dendrogram shows you that. So these are all the sampling sites you saw. Now we're looking at them in the context of these variants we recovered from the environment using that genome. Okay, so we're looking at how they relate to one another. And the first thing you see, there are two major branches. And these branches kept coming up in our analysis, regardless of the distance metric and regardless of the algorithm we use to organize this complex data. And of course, you are curious, where do they fall on the map? And when you look at the map, this is what you see. So those branches look like this. Fine. Because I'm ignorant, I have no idea about oceanography. I was like, wow, maybe, look, all the eastern part of big continents is one color, all the western part of big continents is another color. Maybe it's like high altitude winds pushing some, some fluid dynamics, some weird things going in the atmosphere, and we get more iron always going this way or whatever. An oceanographer colleague of mine said, don't ever say this to anyone. Fine. Here I am telling you. But pretty much it was fun and games. And then later we learned that, in fact, what better explains this is large-scale oceanic current movements. So it seems colder currents are associated with one branch, and warmer currents are associated with the other one. We were extremely excited because we realized that, all right, so there is this huge population with a crazy amount of turnover rate every day. As it goes from a colder current to a warmer one, something happens, and its proteome is selected for certain attributes that make it more able to deal with a warmer environment. So it's like we thought initially that we were certain initially that we were working with a single population, and our conclusion was there are certain variants in the context of its proteome that emerges as a function of temperature. And then we were thinking about how could this happen. There are multiple ways because the population size, maybe the same variants are being invented over and over again as this population continues to move from here to here, and for instance, go, etc. Okay, let me talk about the more subtle variants. So this is two branches, but as you can see, there are some very significant branching patterns here. I cut this tree almost like randomly. I looked at the knee, and it's not a very good analysis, but basically it tells us that there are these things that we can talk about, and when you look where they fall, you start to see some very interesting things. Geographically very distinct part of the ocean, like D here, seems to be identical with respect to their proteome. And when you look at the number of single amino acid variants you recover, you realize that they're extremely fixed. So whatever is going on, we don't know. Maybe it's shipping routes. You know, people go to Antarctic to measure iron in the environment. They can't even make any measurements because the iron that sheds from the ship is enough to contaminate all their, because oceans are iron limited. Maybe that's what's going on. These ships that often travel these routes shed some iron and make the environment iron rich, which selects for a certain proteome in the context of xylem. We don't know any of these. But we know by looking at this, we can tell, okay, 3 minutes. Shh. 3 minutes. But we can tell neutral processes cannot explain alone what's going on with empirical data. So basically. Okay, then we learned that this is not a single population. This happened just a couple of weeks, a week or two ago. We concluded this is not a single population. The paper got rejected from a journal with an excellent question from a reviewer. Thanks to them, we're realizing something much more fundamental to this data, and we're going to go back to it. But it will not change any of our conclusions, but it's important to make the distinction. But don't worry about it. Don't worry about any of it. But what's significant here is that we can look at the merged, for each environment, we can look at a single gene and start talking about what's going on. Very quickly, this is a ribosomal protein. And for instance, purple here shows you amino acids that are exposed to that are not exposed to solvents. And the other two colors are exposed. So purple is buried in the protein structure. And you see in the colder temperatures, you get that one variant that you almost never get in warmer temperatures. So we see this kind of effect of temperature on the proteome. And then here you see things like this. No purple in the tin barrel. The inside of the protein is very fixed. Outskirts of the protein is, okay, purifying insulation leaves you alone. But inside you will not have the folding. And we see things like this that link so well to years of biochemistry research coming from 60 years ago, 40 years ago, is a function of temperature, how protein folding should change with larger amino acids to contain its functioning to be effective. So, and then these, I have colleagues, I know scientists who are taking an amino acid and mutating it randomly to improve its efficiency. So now I can take their amino acid, go to the environment and tell them, well, this guy tells you that don't pet me here. Because if you do something here, the environment has been doing the experiment and it tried enough times for us to know that these two domains are not to be touched, but you can play here, which reduces the search space dramatically for those kinds of experiments. So there's a lot of, I think, this is a better view, biochemical insights. It's been Joe Thornton, a professor from the University of Chicago, who has this beautiful paper from 15 years ago. He says, organismal evolution and biochemistry, these two fields have been distinct from each other, and we need more, interconnection between the two to better understand the mechanistic explanations of organismal level evolution, which I find very inspirational and important. And now I'm realizing that maybe environmental omics data could be the third leg, and we can combine this with approaches from population genetics to better understand these very, very complex questions in the context of oceans or human gut or oral cavity, but to make better sense of what's going on maybe altogether. So we realized the power, and thanks to Evan, who developed carpal tunnel working on this, but he's pushing very hard, we create this interactive platform that you can zoom in, zoom out your proteins in your metagenome to see where your variants fall. You can say, show me all the amino acids that are within 5 angstrom of this particular variant in the tertiary structure. So you can truly get closer and closer to things that are likely to be under positive selection or things that are clearly under neutral processes. Okay, so basically what I'm trying to say is, overall in this talk, that the simulation-based evolutionary model does not really explain all the patterns we're observing when we include data from empirical data from the environment. And there are ways to get to this level of stringency and resolution with current tools that are available to us. So I gave this talk a different version of it without the iPad drawings. At a conference recently, and I was looking how people come up with these cool logos they get funding from. So I had no funding for this. But I decided that it should not stop me from showing cool logos to people. So I thank these guys for their support for my research, being with me, throughout this entire painful process, implementing and everything. And I thank the members of my group very much for their patience. And I thank you for your time, despite that you're all very tired, I can see, in your attention. Thank you. Thanks. So, questions. We have time for questions, right? Couple questions.
When you have the location of an amino acid, how do you know that the open reading frame stayed in the same place? You don't have to mention anything.
Yeah, excellent question. You don't. But if your open reading frame shifted, then the read is very unlikely to map to that context. So what we're doing, the critical part is this. We require full alignment of the read to the genomic context. So if there's insertions and deletions, etc., they are not going to align. And that frame shift will make them fall out from the alignment process. So that's what that's where our confidence comes from. But mapping is a very tricky thing, and I'm sure we're making crap loads of mistakes too. So we also need we can go back into individual short reads, that's what we're doing, and look at those things to come up with better ways of removing noise. We're not perfect at that. Plasmids. Yeah, my lab is becoming a plasmid biology lab, in fact. I'm extremely excited about plasmids. Yes, the quick answer is yes. Long answer, we're just at the beginning. So I don't have anything substantial to say.
So actually, two questions. So she asked you about it's a big issue, the size of the network over the pangenome. It's a big issue because, come on, so, and you know that bacteria is like 4,000, 5,000. So what is the pangenome? And actually, the second question relates to that. So mostly diversity in microbes, probably in ocean as well, is due to lateral gene transfer, and you somehow, okay, I want to hear your...
Yeah, I'm not sure about that. So the questions were, most of the diversity comes from lateral gene transfer in the ocean, perhaps, and how do we start even making sense of these things if that's the case? Second question was pangenomes are very complex, so all those clusters emerging may mean nothing. Correct. And we are very careful about that, but there's one thing going on which is very surprising to me as an ex-computer scientist. There are no heterogeneity continuums in the context of gene sequences. What I mean by that is you cannot just start mutating a gene and then end up with another gene in the genome. It would take so many mutations in bacterial genomes. And this prevents you from merging too many things in those gene clusters. So it's like you see a gene cluster, genes are linking to each other like crazy here, and this is like your Earth, and then the next crazy thing, Saturn, is very far from each other. So even with the worst algorithms, we do rather well identifying gene clusters across genomes very efficiently. So that really, we've been looking at these data a lot, and we're implementing different metrics talk about the homogeneity of gene clusters, both structural and functional. But we have been very happy so far with even our crappy algorithms that are not necessarily suitable for this problem. And lateral gene transfer, we should talk about this later because it will be relevant to many people here. But basically, not really. So
do you have any metadata for the ocean sample?
yes, it doesn't explain. Nothing. Nothing collected, but I think in ecology, oceanography, we don't know how to measure some of the key environmental factors. The things we measure are perhaps not the most relevant ones. Okay, thank you very much.